want me to do that, you can ask. All right, class, so welcome. Here we are, Wednesday, January 26th. I just pulled up our schedule a little bit to show you where we're going. Today, we're talking about cell structures and their functions. So we'll get into the cell. And then on Monday, we'll talk about tissues. Today's PowerPoint is a little bit long, so I'm not sure if we'll get it all through today. So just keep that in mind. It might take us, we might finish it on Monday, in which case your first lecture exam, which is scheduled for a week from today on chapters 1 to 4, it says 100 points, but it will not be 100 questions. We might move this lecture exam to Monday, so just stay tuned for that, depending on how long it takes us to get through chapters 3 and 4. And then in lab for today and Monday, you can see here that we'll be going over exercises 3 and 4, which will be on tissues, but there will be no lab activities. So you can see that your next lab activity actually isn't until lab activity 3, the skin, and that is a Google lab activity. And again, if you have questions on your lab activity, or you have maybe a lab activity you need to make up if you joined the course late, a good time to do that is just to come to your normal lab day. And then when we're done with lab, just ask me, hey, can I get the link from the lab that I missed because I was adding the class or something. All right, so with that, and always, like usual, if you have a question, stay on afterwards and ask me. We're going to jump into chapter 3, which is on cell structures and tissues. And again, we'll see how far we get. Hopefully you've had a little bit of cell biology before, and if not, it's okay. Again, we're in anatomy and physiology, and this is your first introductory course, but we have to lay the foundation a little bit to talk about chemistry and now to talk about cells. So the cell structure is that it has organelles. So the cell itself is its own living unit, and in order to perform all of its specific functions that a cell might need to do, it has organelles. Organelles literally mean tiny organs, and they're specialized structures in cells that perform a variety of specific functions. In lab, you probably practiced labeling the nucleus, the mitochondria, the ribosomes, and then today you'll learn what all of those organelles do. Cytoplasm is the jelly or jello-like substance that holds all the organelles together within the cell. It's just kind of the fluid that holds everything in. And the cell membrane, also called the plasma membrane, is a structure that encloses the cytoplasm. So think of the cell membrane as kind of the border of the cell between the interior of the cell and everything outside of the cell. So that's how the cell is kind of structured. And this is a really popular view of the cell. If you look at this here, you can see the nucleus is kind of our brain of the cell. Within the nucleus, it has its own kind of brain called the nucleolus. And then surrounding the nucleus, we have all our different organelles. We have rough endoplasmic reticulum, smooth endoplasmic reticulum. In green, we have this Golgi Apparatus. We have lots of mitochondria, which are kind of these kidney bean shaped structures with 2 layers inside of them. We have centrioles. They always come in pairs because they will help to divide the cell eventually. We have lots of vesicles. So you'll see lysosomes. These are kind of our garbage collecting parts of the cell, and they will fuse with anything that might come into the cell to try to degrade it and destroy it and get rid of it. We have peroxisomes, which are another type of detoxifying organelle. They help to detoxify a lot of drugs that your cells might be involved or have receptors for. So those are some of the organelles, the structures within the cell. And then you can see here the cell membrane is just the border, the kind of the outer covering of the cell itself. Coming off of the cell membrane, depending on what type of cell it is, we have different kind of things coming off of the cell membrane. We can have cilia, which are really helpful in moving mucus and harmful substances you breathe in out of the body. So cilia kind of help with getting all the junk that you breathe in from the environment. It helps it move it out. And then microvilli are extensions of the cell membrane. They're a little bit smaller, and we'll see those in cells of your digestive tract. to help absorb things that you eat. So you won't usually see a cell with both cilia and microvilli. Those are just illustrated to kind of show you some of the differences of the extensions of the cell membrane. So the functions of the cell itself. So when we talk about cells, what are we talking about? They're the smallest unit of life, so they can reproduce. They're kind of carrying out their own processes within them. They go through cell metabolism and they use energy. And cell metabolism, whenever you see the word metabolism, whether it's by your cells or another tissue, metabolism is just another word for taking in glucose from the food that you eat and turning it into energy. So that's what metabolism is. Cells will help synthesize new molecules like proteins. Cells will communicate with each other, and cells can reproduce and divide and pass on inheritance or the genetic material. And we'll go over a lot of those functions in this chapter. So the cell membrane. So first kind of get through some of these structures. Your cell membrane, again, also known as plasma membrane, it's the outermost component of the cell. It's the boundary or border between material within the cell and then everything outside of the cell. So the plasma membrane is a very important kind of guard, and it only allows certain things to come into the cell. Materials inside the cell, we call them intracellular, and if anything is outside of the cell, we call that extracellular. And the cell membrane acts as a selective barrier, meaning that it lets or allows certain things to come into the cell, but not everything. And we'll see how that does that. We talk a lot about the cell membrane and what makes up its structure, because your cell membrane is really important for, letting things into the cell that could activate that cell to do something. So we'll spend some time talking about the cell membrane first before we go on to the other structures. But we use the terminology fluid mosaic model, and we use this to describe the cell membrane structure because it's a mosaic, it's made up of a lot of different pieces, and it's made of this fluid form where all the pieces are kind of moving within it. The membrane itself contains phospholipids, which we'll go over how those are formed, cholesterol, proteins, and other carbohydrates. And the phospholipids themselves form a bilayer, which just means we have two layers of phospholipids that kind of form this cell membrane structure. And the phospholipids themselves contain a polar region and a non-polar region. And we'll talk about what that means and when we show you a picture. So the phospholipid molecule has a polar, what we call head, that is hydrophilic. Philic means love, so the polar head loves water, and the nonpolar tail is hydrophobic, and phobic is afraid of water. So the polar head or the polar regions will always be exposed to water around the membrane, and the nonpolar regions will be facing the interior because they don't want to touch water. And I'll show you a picture of what I mean by that next. So here we have the cell membrane structure, and I'm just going to focus your attention on the phospholipid bilayer first. So it's two layers of these phospholipids. You can see a layer at the bottom and a layer at the top. And the blue kind of head is polar hydrophilic, so it loves water. So the heads of these phospholipids will always face the exterior of the cell up here where there will be water, and the phospholipid, the second layer, kind of polar head, will face inside the cell where there will be water. But you can see here that the non-polar regions are the tails. They look like pieces of spaghetti touching each other. And the non-polar regions are hydrophobic, meaning they do not like water. So they will touch each other because then they'll just only be in contact with each other and never have to touch water. So that's how we get this phospholipid bilayer and how it rearranges itself, where the polar heads face inward and outward and the tails face just into each other. Also making up the cell membrane, we have different channel proteins and receptor proteins. So here's a membrane channel where it would allow things to get in and out of the cell. We have other proteins. These are carbohydrate chains, like chains of glucose molecules that are reaching out from outside the cell. We have receptor molecules, which will bind to some sort of molecule. And when anything binds to a cell on the cell membrane, that will start a signaling process on the inside that signals to that cell what to do. So a lot of your hormones, a lot of drugs, toxins, actually, they will either enter the cell directly, and if they're not able to enter, they'll bind to some sort of receptor on the cell membrane. And once that drug, hormone, or toxin binds, then a series of reactions will occur inside the cell to say, hey, thyroid hormone just bound to us, let's start dividing. Or hey, nicotine, the drug just binded to us, this is the response that we'll get. So that's why receptor molecules are important. They serve as kind of a receptor for molecules that cannot pass through and get into the cell itself. And then on the inside of the cell, we see different cytoskeleton fibers. So just like our bodies have bones as our skeleton, your cells have different fibers, which make up the cytoskeleton, which kind of give the cell its shape and provide stability for it. All right, so we're going to keep moving then. So movement through the cell membrane. So I talked a lot about how some things cannot go in. And again, that's what we call selective permeability. So the cell membrane only allows certain things to pass in and out of the cell. Some substances like enzymes, glycogen, which are long chains of sugar, and potassium are always found in higher concentrations inside the cell. And then substances like sodium, calcium, and chloride are always found in higher concentrations outside of the cell. And those kind of concentrations means how much there are of them. We'll come back to this when we get to your nervous system and see why things like potassium are always in a higher concentration inside the cell, and things like sodium are always in a higher concentration outside of the cell. So we'll come back to that when we get to nervous system to kind of see why that is. Some substances like oxygen and carbon dioxide can pass directly through the cell's membrane's phospholipid bilayer because they're also non-polar, so they can pass through. Some substances must pass through transmembrane protein channels like sodium. So an example of one of these transmembrane sodium channels is shown there. So those channels are for polar molecules that might have a charge to help them get across the membrane. And the route of transport through the membrane, it always depends on the size. Usually smaller substances can get across the membrane more easily. The shape, if something is too big and bulky, it won't be able to pass through. And then the charge, if anything is polar or has a charge, like positive sodium, it needs a protein channel to help it get through as well. Some substances require a carrier molecule to transport them across. So glucose, for example, will kind of piggyback off of another molecule to help it get in the cell. Some substances require what we call vesicular transport, where they will actually kind of be within some sort of vessel or sac, and then that vessel will get ingested into the cell. The vessel kind of will fuse with the cell membrane to enter or exit. And then we talk about terms active and passive transport. And transport is describing things going into or out of the cell. And these transport processes can be passive, which do not require the cell to expend any energy. So passive membrane transport will occur when things can just, passively, easily flow across the cell membrane into the cell or out of the cell. So the cell doesn't have to kind of push that vessel in or push it out or pull it in. There's no energy required for the cell to transport these molecules in or out. However, when we talk about an active membrane transport process, this does require the cell to expend energy. And again, energy in our body is always in the form of this really fun molecule called adenosine triphosphate, abbreviated ATP. And think of an active membrane transport process as one where the cell has to like pull something in, use energy to do that, or push something out, which requires energy. But a passive transport process, things will just come in and out freely without the cell pulling or pushing them. Some active and passive transport processes then, that we have a lot of them, because again, it's so important to kind of understand how things get into and out of the cells, because this is how your cells will respond to a hormone or a drug. So that's why we spend time just talking a little bit about how things come in and out of the cell. So passive membrane transport mechanisms, which again happen freely, no energy is required by the cell for these to happen, are what we call diffusion, osmosis, and facilitated diffusion. And an active membrane transport mechanism, which again means the cell is either pulling for something to come in, pushing it, or another mechanism is helping it come in. These include what we call active transport, secondary active transport, endocytosis, and exocytosis. And we'll go over these a little bit now. So diffusion, this is kind of the simplest one. Diffusion generally involves what we call substances moving down their concentration gradient. So that means that some sort of substance will move from an area where it's more highly concentrated to an area that there's not that much of it. And diffusion, a really simple way to describe diffusion is this happens when you spray perfume in a room. At first, when you spray that perfume, it's just in the one area where you sprayed it, but eventually that perfume will diffuse to all areas of the room. So that's diffusion, where those perfume molecules are moving from an area of highly concentrated to lower concentrated. We talk about a solution is generally composed of two parts, a solute and a solvent. And it's important to define these three terms because we'll use them to describe diffusion and how this takes place. But a solute is any substance that can be dissolved in a predominant liquid or gas. So for example, our solute might be the perfume particles that we sprayed in the air that are dissolving or spreading out in the gas. The liquid or gas then is called the solvent. And another example of solutes and solvent would be taking a glass of water, which would be solvent, and pouring some salt in the water. The salt would be the solute. And eventually, if you stir up the salt enough, the solute of salt will become dissolved and spread out or diffuse into all areas of the water, which is the solvent. And when we combine the solute and the solvent together, that's where we get a solution. And we get that solution of salt water, for example. The solutes themselves, so for example, my example of salt or the perfume, any ion or molecule will always tend to move from an area where it's more highly concentrated at first, and then over time, all the particles will spread out to an area where they're more lower concentrated of that same solute in a solution. And that's just because naturally these particles are kind of bouncing off each other and they want to get spread out until there's kind of an equal concentration of those solutes within the solution. Another example, of this diffusion idea is putting creamer in your coffee or sugar in your coffee. Putting creamer in, you eventually see the color of the coffee change to more of a milky brown color. So that creamer is diffusing through all parts of the coffee. Or putting sugar in your coffee. The sugar is the solute, you mix it up, and that sugar moves from an area where it's more highly concentrated to all areas of your cup of coffee where it's more lower concentrated. So that's this idea of diffusion, when any solute moves from an area of high concentration to low concentration. Maybe that made you want to get some coffee, I don't know. I know it's early where you guys are at. The concentration gradient then itself is just the difference in the concentration of a solute in a solvent between 2 points divided by the distance between the 2 points. So I'll talk about the concentration gradient and we'll say it is steeper when the concentration difference is large or the distance is small. So for example, as we look at diffusion here, And this is an example of putting a thing of salt, a salt crystal, which is the solute, in a solvent of distilled water. And you see that the salt ions will move down their concentration gradient into the water as they're completely dispersed throughout the water. So that's the salt diffusing through all parts of the water until eventually the salt ions are evenly distributed, water molecules are evenly distributed, and we have this nice saltwater solution. So that is diffusion. How does this relate to cell membranes? Well, depending on where a concentration of a solute is greater, for example, outside the membrane or inside the cell, when a channel opens, a channel protein, for example, that solute or whatever molecule you're talking about will always move from an area where it's more highly concentrated into an area, for example, inside the cell where there isn't as much of it. So that's simple diffusion. It doesn't require any energy to occur. Leak and gated channels, so anything that's lipid soluble can diffuse directly through the phospholipid bilayer, because remember the tails, the inside of the bilayer has those nonpolar tails. They don't like water. So if you have a lipid soluble substance, which also does not like water, it will easily diffuse through that phospholipid bilayer. But if you have anything water soluble, such as an ion, they can only diffuse across the cell membrane by passing through a channel. And we have two kind of types of channels that are leak channels that constantly allow ions to pass through. So think of leak channels, they're always open. Ions are constantly kind of leaking through these leak channels. A gated channel, however, limits the movement of ions across the membrane by only opening and closing in a certain situation. So a gated channel will have a gate that only opens or closes to allow ions to pass through. So those are leak and gated channels. So this is a look at diffusion through the cell membrane. Again, to make this really easy, you see the concentration gradient downward in this direction, meaning that there's a higher concentration of these red, green, and yellow molecules above the cell membrane or exterior to the cell than on the inside. So that's why the concentration gradient has an arrow showing the downward arrow, because that shows the direction that those molecules will diffuse to an area where there aren't as many of them. Lipid-soluble molecules, again, like fats, they are also hydrophobic. So they can pass directly through the cell membrane, because remember the inside, these tails are also hydrophobic. So lipid-soluble molecules can pass directly through, but non-lipid soluble molecules cannot pass through. So if we have a specific non-lipid soluble molecule or ion that has a specific channel that it's associated with, a lot of times there's only specific channels that will work with specific ions. When this membrane channel opens, or it's a leak channel and it's constantly open, that's when these non-lipid soluble molecules can pass through, because they're bypassing, they're not going through the tails, they're going through the membrane channel to get inside the cell. So that is diffusion. Passive transport process does not require energy. Here's an example to look at our leak and gated channels. Leak channels, again, are always open. This shows an example of a potassium leak channel that's always open. We'll get to this when we talk about nervous system. And then a gated channel, here's a gated sodium channel closed and a gated sodium channel open. And you can see how it has a gate here that will open and close. And when it opens, that's when sodium can pass through. And again, just keep this picture in mind, because we'll come back to it when we get to your nervous system as well. Okay, so are we ready for another transport process? Are you still with me? The next transport process we have is osmosis, which is another passive transport process, so it does not require energy. It really simply put, osmosis is the diffusion of water or the diffusion of a solvent. Thanks for the message in the chat. I'm glad you guys are still here. So osmosis, we talked about diffusion of ions and molecules. but osmosis is just the diffusion of water. The movie Osmosis Jones. I haven't seen that movie. What's that about? I haven't even heard of it. You can tell I've not been to the theaters in a while, but I did see Encanto. Have you guys seen Encanto? You probably only have seen it if you have little girls like I do, but it's a great movie. Okay, Osmosis Jones. Thanks, Ruby. So osmosis is the diffusion of water. It's a solvent across a selectively permeable membrane from a region of higher water concentration to a region of lower water concentration. And osmosis, or this kind of push from water, always exerts a pressure, which is what we call osmotic pressure, which is the force required to prevent movement of water across the cell membrane. And did you have a question, Ruthie? I just saw you raise your hand.
Yeah, sorry, I don't mean to interrupt, but I mean, I highly recommend it. It explains everything you're going through with this. It's an old cartoon, actually.
Awesome. So it's an older cartoon that talks about osmosis.
Yeah, I think you can find it on YouTube. It's
Very
not that
cool.
long.
Okay.
Yeah.
Thanks, Ruthie. I've never heard of that. Thank you so much for bringing that up.
No problem. Sorry to interrupt.
No, that's no problem. Thank you. All right. So osmosis can exert a pressure called osmotic pressure, which is the force required to prevent movement of water across the cell membrane. And we'll kind of show you what this means when I go over some pictures. But osmotic pressure depends on the difference of solution concentrations inside of the cell relative to outside of the cell. And this will be really important. So we're going to kind of dive into some details. It'll get a little complicated, but this will be very important when we're talking about giving someone an IV or a saline solution to make sure it's the right concentration so that that person's cells will not explode or shrink up in that solution. So that's kind of where we're going with this. So a cell can be placed in a solution that's either hypotonic, isotonic, or hypertonic compared to the cell cytoplasm. What that means is the cell can be placed into a solution. Let's say I put a cell into my water bottle here. And when I put my cell into a hypotonic solution, the water inside my water bottle will be less concentrated. Hypo means less than the concentration within the cell itself. If I put a cell into an isotonic solution, iso means same. So we're placing a cell with has an inside concentration of something. We're putting it into a solution that also has the same concentration relative to that cell's cytoplasm. And if we put a cell into a hypertonic solution, hyper means more. So the solution that we're putting the cell in is greatly or more concentrated compared to the inside of the cell cytoplasm. So this kind of goes into a little more detail. Again, a hypotonic solution has a lower concentration of solutes and a higher concentration of water compared to the inside of that cell. The solution has what we call less tone or less tonicity or osmotic pressure than the cell itself. So this is really important. What happens when we put a cell into a hypotonic solution? Because the inside of the cell is more concentrated than the solution itself, water moves by osmosis into the cell. Remember that water moves by osmosis from anywhere to a higher water concentration or a hypotonic solution to a lower water concentration. And when this happens, water will move into the cell, causing it to swell. And it could eventually go through a process called lysis, where it actually ruptures or splits apart. So that is placing a cell into a hypotonic solution. And to remind yourselves, or a little trick to remembering this, is when you put a cell into a hypotonic solution, exaggerate this O, and the shape of that O is what happens to the shape of the cell, because as you place a cell into a hypotonic solution, water moves into the cell, causing it to swell and maybe lyse. So I'm just going to put H2O there as it's showing it moves into the cell. So that's hypotonic. And eventually, I think I show you a picture of this too. An isotonic solution is the best case scenario because iso means same tonicity or same concentration. So if we put a cell in an isotonic solution, the inside of the cell and the outside of the cell are equal in concentration of their solutes. So this means that water, might move freely in and out of the cell, but there's no net movement of the water. So the cell will neither shrink nor swell. These isotonic solutions are what we have figured out to be kind of normal saline solutions that we give to people who've lost a lot of fluid. We always want to give someone an isotonic solution so that when it bathes their red blood cells aren't shrinking or swelling because they're in a happy environment where the inside of their cells are in the same concentration as the solution they're placed in. And then a hypertonic solution. Hyper means more. So when a cell is placed in a hypertonic solution, the solution itself has a greater concentration than the cell. So the cytoplasm of the cell in a hypertonic solution has a lower solute concentration and a higher water concentration in the surrounding solution. So water, again, will always move to an area where there's kind of less water. So water will move by osmosis from the cell and into the hypertonic solution where there's a greater concentration. And that will result in cell shrinkage, and we call cell shrinkage crenation. So again, we put a cell into a solution where it's more concentrated, so water will naturally move into that area of more concentrated solution. When water leaves the cell will just shrink up, shrivel up, and we call that crenation. So here is like a very typical experiment that we show what osmosis is. And again, osmosis just means the diffusion or the movement of water, where water will always move from an area where there's more water molecules to an area where there's less water. Another way of saying that is water will always move into an area where there's a greater concentration of solutes. And sometimes I tell students, if you can remember the word or the phrase solutes suck the water into them. And that's the way water travels, by osmosis. So this really kind of famous experiment is we take kind of a beaker that's covered at the end with a selectively permeable membrane. That means that only water can get through this membrane. Whatever is inside in this green fluid, whether that's salt or sugar, cannot get out. So we have a 3% salt solution. So it's filled up with salt crystals and a selectively permeable membrane at the bottom. And we kind of submerge this kind of hammer or beaker into another beaker that's filled with distilled water. And distilled water has like a 0%. There's nothing in it. So it has nothing in it. So we're putting this salt solution into an area where there's, 0% of anything concentrated. And you can see here that through osmosis, water will travel into an area. And you can see this here, you can see the arrows of water traveling into that salt solution because water naturally wants to go to an area where there's more solutes. And that's because water naturally wants to create kind of an equilibrium between concentrations. So water travels into the salt solution via osmosis. And at the end, after water has moved in, you can see that the weight of the water column, so at first you can see the salt solution starting to rise, and at the end you can see it rising even more as water is moving in to this kind of funnel. So hopefully that makes sense a little bit. That's the movement called osmosis. It's the movement or diffusion of water. And this is also a passive process, so it does not require energy at all. And this is a picture that I was describing before when we placed red blood cells into the 3 different types of solution. So let's look at the isotonic solution first. So a normal red blood cell has this nice kind of donut-like shape to it, a biconcave disc. And when we place a red blood cell into an isotonic solution, iso means same concentration, where the inside of the red blood cell has the same concentration as the solution that it's placed in. You can see here that water moves freely into and out of the cell, but there's no net movement of water because the concentrations are the same. If we put a red blood cell into a hypotonic solution, remember, emphasize the O, because that tells you the shape of the red blood cell will take when you put a red blood cell into a hypotonic solution. Because the inside of the red blood cell is more concentrated than the solution itself, water will naturally flow into that red blood cell, causing it to swell and eventually burst or lyse. Yes, Ruthie, did you have another question?
No, sorry, oops.
Oh, no problem. I know sometimes the hand, yeah, no worries. And then if we put a red blood cell into a hypertonic solution, and again, hypertonic describes the solution, the water around the cell is more greatly concentrated. Water by osmosis will diffuse out of the cell and the cell will shrink up or shrivel up, and that's called crenation. Okay, so hopefully that gives you a little more information about osmosis. So we're going to talk a lot about transport, I told you, but eventually we'll get to some of the other organelles in the cell. Carrier-mediated transport. So some water-soluble, electrically charged, or larger particles cannot enter or leave the cell membrane by diffusion. So these can include things like big amino acids, glucose molecules, and some polar molecules produced by the cell. And these carrier molecules are proteins within the cell membrane, and they're involved in carrier-mediated transport. So again, a carrier molecule is a protein in the membrane that just helps kind of carry a molecule across the membrane that needs help to do that. Carrier-mediated transport mechanisms can either be facilitated diffusion, which does not require ATP for energy, or active transport, which does require ATP for energy. So these are kind of two types of transport mechanisms to get something across the membrane. Facilitated diffusion does not require ATP, but active transport does. And we'll go over these now. So facilitated diffusion will use some sort of carrier, or think of it as like a piggyback person, to get a substance across the membrane, and it will always go from an area of high to low concentration of that substance, because this will not require energy. So ATP is not required. So these substances will always flow down their concentration gradient and no energy is required. The only thing that is required is some sort of carrier, like a backpack or a piggyback ride, for these substances to get across. So a big example of this is glucose. So glucose cannot pass the cell membrane by itself, but all your cells need glucose because inside the cell, glucose is what is used to create more energy, or ATP. So glucose has to get in the cell, but since it's such a big molecule, it cannot cross the plasma membrane by itself. So it uses a carrier molecule that kind of just helps it across. But glucose will always travel from an area where it's more greatly concentrated to an area where there's not that much inside the cell. So it'll flow down its concentration gradient. And because it does that, even though it's using the help of a protein carrier molecule, we still call it a type of diffusion that does not require energy. So no ATP is required to get glucose across. It just flows down its concentration gradient using the help of a carrier molecule. Active transport also uses a carrier molecule, but it requires ATP, and it always moves a substance across the cell membrane, but against the concentration gradient. So it moves something across, for example, an ion or a molecule from a lower concentration to a higher concentration against the concentration gradient. So think of this as you're trying to push something uphill, you need energy to do that. I also describe this, if you ever ride the buses or a train that are jam-packed full and you're trying to get into a bus, you're going from an area lower concentrated and you're trying to push your way into an area of higher concentrated people. And that requires energy. You have to push against to try to get into that bus or train. Active transport processes will accumulate necessary substances on one side of the cell membrane at concentrations many times greater than those on the other side. And the big example of an active transport kind of pump or process is called the sodium-potassium pump. And we are going to spend a lot of time talking about this when we get to our nervous system. But this is a major example of active transport that uses or transports sodium and potassium ions in the cell membrane. And this kind of pump always moves sodium out of the membrane or out of the cell against its concentration gradient, and it moves potassium into the cell. And what this pump does is it always produces a result of a higher concentration of sodium outside the cells and a higher concentration of potassium inside the cells. And this pump is incredibly important because without this pump, your nervous system would not function. These concentrations of sodium and potassium and making sure you have sodium outside the cell and potassium inside of the cell are required for your nervous system to function. And that's all I'll say, but hopefully that gets you excited to learn about the nervous system. So here's the sodium-potassium pump. Really simply put, it's a carrier molecule protein embedded in the membrane, and it transports 3 sodiums out. So you can see those 3 sodium molecules out at the same time that it transports 2 potassium ions in. And you can see here how ATP kind of also attaches to this membrane. It breaks a bond, so it breaks off a phosphate group. And when it breaks that bond, that provides the energy to do this kind of transfer of 3 sodiums out and 2 potassium in. So that's the sodium-potassium pump. It's an active transport process. It's moving these ions against their concentration gradient, and the result will be to establish a greater concentration of sodium ions outside the cell, so above the cell membrane, and a greater concentration of potassium ions in the cell membrane. And again, that's super important for our nervous system to function well. Okay. More transport processes. Everyone loves this, but I promise it'll get more interesting. So bear with me. Secondary active transport also uses energy provided by a concentration gradient that is established by an active transport of one substance, like sodium, to transport another substance. No additional energy is required above the energy provided by that initial active transport pump. And we're going to talk about kind of 2 types of secondary active transport. In co-transport, some sort of substance diffusing will move in the same direction as the initial actively transported substance. And in counter-transport, the diffusing substance moves in a direction opposite to that of the initial active transported substance. So a good example of this, we have secondary active transport. So the sodium-potassium pump is an active transport mechanism. So that requires energy to move sodium out of the cell and potassium into the cell. But then the secondary active transport mechanism does not require energy, because all it's doing is this is a co-transport process, a secondary active transport. What it's doing as sodium moves back into the cell down its concentration gradient, that is the energy that helps to bring in glucose at the same time. So that's why it's called secondary active transport or co-transport. Kind of when sodium moves into the cell, that provides the energy required to move glucose, for example, into the cell against its concentration gradient. So this is an example of co-transport, a secondary active transport process. Endocytosis and exocytosis. Endo means into, so this is moving something into the cell. It brings a material into the cell using a vesicle, which is like a sac or a bag that moves things into the cell. Receptor-mediated endocytosis occurs when a substance will bind to a receptor and then transport it into the cell. Phagocytosis is often used for endocytosis when something solid is ingested by the cell, so the cell actually eats something. And pinocytosis is reserved for much smaller vesicles formed, and that contain liquid rather than solid. So pino means to drink. So think of pinocytosis as the cell drinking a liquid in, and phagocytosis is the cell ingesting something solid into the cell. And this is a look at receptor-mediated endocytosis, where a molecule that needs to get into a cell will bind to a receptor on the cell membrane. The receptor and the bound molecules will then kind of form this vesicle that can then go into the cell. So that's receptor-mediated endocytosis. And then exocytosis, exo means out of the cell. This involves the use of a membrane-bound sac called a secretory vesicle because it's being secreted from the cell. And this is used for materials that have been accumulating inside the cell, maybe wastes, toxins that the cell needs to get rid of. And the vesicles move to the cell membrane, they fuse, and then they're ultimately release the material by what we call exocytosis. And a good example of exocytosis is the secretion of our digestive enzymes. So we have specific cells in our digestive tract that secrete lots of enzymes used to help us digest our food. So these cells make the enzymes, within the cell, but then in order for those enzymes to get out of the cell, they'll put those enzymes in a vesicle and kind of spit it out. So that digestive enzyme can then go on to your digestive tract to help break down your food. So here's a look at exocytosis. We have a secretory vesicle with vesicle contents inside of it. The vesicle will fuse with the cell membrane and then actually spit it, or kind of like vomit out these contents inside that vesicle. And this is a transmission electron microscope at about 30,000 times magnified of a cell undergoing exocytosis. And you can see these contents are just being spewed out of that cell. A pretty cool picture. All right, so now we get into general cell structure. Let me check the time. I'll go for about 10 more minutes. I know we technically have a class for an hour and 20 minutes, but after an hour, I don't know how much our brains can handle. So I usually never lecture for more than an hour. So we'll get through maybe 10 more minutes of slides, and then I'll give your brain a chance to relax a little bit. But we'll continue this. If we don't finish this PowerPoint today, we'll finish it on Monday, which might push off our lecture exam, but I'll keep you posted about that. So now we'll talk more about the general cell structure, and we'll kind of begin our discussion about the organelles inside the cell. So the interior of your cell is composed of the cytoplasm, and that is all of the jelly-like fluid that surrounds the organelles. And organelles, which I talked about at the beginning, are a specialized structure. Think of an organelle like a tiny organ inside the cell that performs a specific function. And these are a list of all the organelles inside the cell. In lab, you labeled some of these or practiced labeling some of them. And now we'll kind of give you more of a detailed look at what all these organelles do. So this is the picture I showed you, kind of one of the first couple slides. This is a great kind of generalized cell structure that you'll see in a variety of textbooks, no matter what you study or which textbook you study cell biology from. This is a pretty popular image that they use. You'll see extensions from the cell membrane here, the microvilli, the cilia, and then within the cell are all of these organelles, and we will go over their functions. You can see a little vesicle undergoing exocytosis here, so you can see that occurring. So we'll start with the nucleus, and the nucleus is like the brain of the cell. The nucleus is a very large organelle. It's usually located in the center of the cell, and it's bounded by what we call a nuclear envelope, which is kind of like a membrane to the nucleus itself. And the nuclear envelope consists of outer and inner membranes with a space between them. And what's also important about this nuclear envelope or nuclear membrane, it contains holes called nuclear pores, through which materials can pass into and out of the nucleus. And these pores, really specifically, your nucleus kind of keeps all your DNA and RNA, all your genetic materials inside the nucleus. And when your DNA gets replicated or a copy is made, it can leave the nucleus via these nuclear pores. The nuclei, so that's plural for nucleus, of human cells contains 23 pairs of chromosomes, which consist of DNA and proteins. And during most of a cell's life, the chromosomes are loosely coiled, and we collectively call kind of your DNA and proteins chromatin because they're loosely coiled as chromosomes within the nucleus. But when a cell is about to divide, so if you take one cell and you divide it into two, that's when the chromosomes become very tightly coiled into the kind of their X-like structure that you might have seen a chromosome having. And then you can see these chromosomes underneath the microscope. The DNA becomes tightly coiled or wound into chromosomes because a chromosome with the tightly wound DNA, the X shape, is much more easy than to break apart that DNA to make sure you're passing along half the DNA to each new daughter cell that's formed after a cell divides into two. Within the nucleus are the nucleoli, which are what we call diffuse bodies with no surrounding membrane that are found within the nucleus. There's usually one to several nucleoli within the nucleus, and the subunits of ribosomes, which are a type of organelle in your cell, are formed within the nucleolus. And we'll talk about what ribosomes do, but ribosomes are formed within the nucleolus, and then they kind of leave or exit the nucleus through their nuclear pores. So you have subunits, and ribosome will have maybe like 3 subunits that are made, and each subunit will leave or exit the nucleus through the nuclear pores because it'll be smaller to get out that way. And then those 2 or 3 subunits of the ribosome will then come together to form a complete ribosome when it's outside of the nucleus. And we'll talk about what ribosomes do. So here's the cell nucleus. You can see the inner nucleolus, and this is a nice picture of the nuclear envelope. And you can see here the kind of inner and outer space, and you can also see the pores or the holes that again are just openings that are moving into and out of the nucleus itself. And again, those pores are important for genetic material to pass through. They're important for those subunits of ribosomes to pass through. Here's a look at chromosome structure. So within the nucleus, normally your DNA, it's this kind of helical shaped structure, contains all your genetic material. It winds itself around proteins, and DNA and proteins are just loosely kind of packed together during most of a cell's life. But during or right before cellular division, these chromatin or the DNA will then become even more tightly wound to form a chromosome. And a chromosome is kind of this X-like structure of very tightly wound DNA and proteins. And again, DNA will kind of form this chromosome shape right before the cell divides, because as a chromosome, it's easier to pull apart a chromosome right here. It's called the centromere. It's easier to pull apart a chromosome when it's tightly wound into this shape. Okay, so we jump right into ribosomes. I promised you I'd stop at in 1 hour. Ribosome components are produced in the nucleolus, what we said, and then ribosomes specifically are the organelles where proteins are produced. So if you remember anything today, pick out keywords or phrases, big picture, ribosomes make proteins. That's all you need to remember about ribosomes. Ribosomes make proteins. Ribosomes can be attached to another organelle called the Endoplasmic Reticulum, where they're kind of studded in that organelle, or ribosomes are freely floating in the cytoplasm, and we call them free ribosomes. So ribosomes create proteins, and they'll either be attached to another organelle called your endoplasmic reticulum, or they'll be freely floating throughout the cell, and then we call it a free ribosome. Here is just a look at ribosome production. You can see kind of subunits of ribosomes being made in the nucleolus. You see these subunits leaving through the nuclear pores, and then kind of a complete ribosome forming here. And this ribosome will eventually, it's showing what we call an mRNA strand kind of running through it, because this mRNA strand are kind of directions for making a protein. So the ribosome will read those directions and then make a protein off of those directions, because ribosomes create proteins. Just get that into your head. Endoplasmic reticulum, so we talked about the nucleus, ribosomes creating proteins. The endoplasmic reticulum is then our next organelle, often abbreviated ER. It's a series of membranes forming sacs and tubules that usually extend from the outer nuclear membrane into the cytoplasm. So we'll always see the endoplasmic reticulum kind of surrounding the nucleus. The rough endoplasmic reticulum is called rough because it has attached ribosomes to it. So I told you ribosomes are sometimes attached to the endoplasmic reticulum. If the endoplasmic reticulum, if the ER has ribosomes attached to it, we call it rough ER. And that ER will be involved in protein synthesis because ribosomes create proteins. The smooth ER is nice and smooth, so it has no attached ribosomes to it. Your smooth ER is more important for lipid synthesis, so making fats, cellular detoxification, so detoxifying anything harmful that your cells might be exposed to, as well as storing calcium ions in skeletal muscle cells. So you'll find actually a lot of smooth ER in cells in your liver, because your liver is the big organ for detoxifying anything harmful you take into your body, whether it's Tylenol or whether it's alcohol. So you'll find a lot of smooth ER because its purpose is to detoxify the cell. You'll find a lot of smooth ER in cells of the liver. So here's a look. If you kind of look at this picture first, you can see, and I like how they always gray out the organelles that you don't need to focus on, but I'm just going to circle or focus on the nucleus. And then you can see how what's been highlighted is the endoplasmic reticulum. And then if you zoom in on it, you see how the rough endoplasmic reticulum is kind of a continuation of the nuclear envelope from the nucleus. And then the smooth endoplasmic reticulum is a continuation of the rough ER, but without ribosomes. So your smooth ER does not contain ribosomes, and your rough ER does. And you can see these little kind of circles attached to the rough ER. Those are the ribosomes that are kind of studded or embedded within those membranes. Okay, we'll talk about Golgi and then we'll pause here for the day, but your Golgi apparatus, also called the Golgi Complex, and you'll probably see those two words interchanged either in your lab material. So just keep that in mind. The Golgi apparatus is also called the Golgi Complex. It also consists of closely packed stacks of curved membrane-bound sacs. And the big thing that your Golgi does is it collects, it modifies, it packages, and distributes all the proteins and the lipids that are made or manufactured by your endoplasmic reticulum. So we talked about how rough ER creates proteins and smooth ER creates lipids. Well, once they're made by your endoplasmic reticulum, they're then sent to your Golgi. The Golgi apparatus is like an Amazon distribution center. It will collect items, it modifies them, packages them, and distributes them out. Also think of the Golgi apparatus like a post office. It will take all the packages and then distribute or send them out where they should go. The Golgi apparatus can form vesicles with, the protein or lipid that's inside of it. Some will be a secretory vesicle, which will be spitted out of the cell for secretion. Some might be a lysosome, or some might be some sort of other vesicle. So that's the Golgi apparatus. You'll see it often, colored in green in cells. I don't think it necessarily is the color green, but many textbooks color it green. And you'll see here how one end kind of is the receiving end for all the materials, and then one end will be the secreting end, where it might create a vesicle that's secreted out of the cell. So that's the Golgi apparatus. It's like your post office. It takes everything that's been made by the endoplasmic reticulum and then gives it a destination of where it should go. So I'm going to pause there and I'll make a note that we'll start on lysosomes. And I'm going to go ahead and stop the recording and stop my share. And if you have any questions,